want, Tracy, you want to get started, and then the three of us will mute ourselves.
Yes, and I will do the intro, and then I'm going to mute myself because I don't need to talk after that. So I first wanted to welcome everyone here to the Atorvastatin Clinical Trial update. I'm Tracy Brown. I am the engagement specialist with Angioma Alliance. One of the best parts of my job is that I get to bring people together with other affected families. In 2011, my son had a brainstem hemorrhage, and the first thing I did was get on the internet and see what I could find out. And then I found someone who had it, and I took my son to meet him because being rarely diagnosed, I didn't want him to feel alone. That's really scary. The next thing I did was plan a fundraiser to help fund research because they said there's nothing, it's not operable and there's nothing you can do. And I said, well, that's silly. Of course, we've just got to figure out what to do then. So we started the Zach Brown 5K, and that's been real successful, and other people have done other fundraisers after finding us online. So I'm real proud of that. The Angioma Alliance is working to create smaller community groups. There are, I should know this number, but I think 8 state groups currently, and we're starting more every day. So if you are interested in meeting up either with other families or individuals, just for activities or for fundraising events, awareness events, I will make it easy for you. All you have to do is reach out to me and tell me that you're interested, and I will take it from there. So you can reach me at tracy@angioma.org or just find me on the website. And I'm going to introduce Joanna Jimenez. She's the chairperson for the Florida Angioma Community Alliance, and she's going to be guiding the event tonight. There you go.
Thank you, Tracy. Hello. So I had a seizure at the age of 13, and I was operated at the age of 16. It's been about 15 years since, and I must say I don't get too many symptoms, but I did want to give back to the community, and so I became a chair member last year. And as the Florida chapter, we wanted to create awareness and host, thanks to technology, to spread news. So we figured a webinar would be ideal, and one of our members, Dr. Awad, should tell us more about his clinical trial. So this is actually the second one we host, and we're very excited about this. So now let me introduce to you the reason we're all here. And he is the president he's internationally recognized leader in neurosurgery. He leads the neurovascular surgery program at the University of Chicago. He is a pioneer, a scholar, and he is definitely leading the changes in his industry, especially with the first ever clinical trial for the angioma, which is why we're all here. And let me introduce you now to Dr. Awad. Before I bring him on, I would just like to say we will have questions and answers at the end. There is a Q&A button in the bottom of your screen where you can answer questions. If you've already submitted your questions, we have them and we will go over each one. So let me introduce Dr. Awad now.
Thank you, Joanna. Thank you, really, the whole Angioma Alliance for leading this. You guys are fantastic, and you are our inspiration every single day for what we do. So this webinar is about the first clinical trial that is being launched for a cavernous angioma that has bled, that has caused the symptomatic hemorrhage. And it is designed to test the effects of a drug called Atorvastatin. And the reason this drug is being pursued is because it has effects on the cavernous angioma that stabilize the bleeding and stabilize the growth of the lesion. This was discovered in the laboratory after about 10 years of very serious experiments. And eventually, we developed a way on the MRI of the brain to measure the bleeding in the lesions. And we proposed to the National Institutes of Health that we test this drug for the first time in human patients with cavernous angiomas. This drug is approved by the FDA for cholesterol-lowering effects and for the prevention of stroke and heart attack, but it is not approved for cavernous angiomas. So we have to show that it is safe and effective. And the way we are proposing to do that is to take the patients who have had a symptomatic hemorrhage in the year prior to enrolling. So they could have had their bleed up to 12 months prior to coming to us, and the lesion must not have been already removed surgically or radiated. So we don't want a lesion that's been operated or radiated. So if that lesion that bled is still there and is being followed, as we know many cases are, then the option is for you to consider enrolling in the trial. And we're going to spend some time tonight explaining how the process of enrolling in the trial plays out. And it is going to basically tell you how you go about getting in touch with us, how we can assess whether you are eligible for the trial, and how we can explain it to you. I am basically going to be the question and answers guy tonight for most of the webinar, and I'm going to actually have three different young ladies talk to you first. One on video to tell you about her experience in enrolling in the trial. And then we're going to have Christina Paedad, who is the trial nurse. And many of you may know her because she is the CCM nurse at the University of Chicago. And she's going to explain to you how it goes about from the time you call us or email us, and how we assess whether you're eligible and so forth. And then we are going to have Agnieszka Stadnik, who is our clinical coordinator, explain about the consent and the regulatory processes and how we assess the compliance with the trial. And then finally, before we go to the questions and answers, I'm going to explain to you a little bit how this trial fits in with your regular clinical care. Like how do you continue to receive your care by your regular doctors even when you are in the trial, because this is always a question. Then the rest of the information about the trial, there is a brochure that we gave you that basically explains the general lines of the trial and who is eligible, but more importantly gives you numbers and email contacts very specific to this trial. So you can email at cash@uchicago.edu, for example, and this is an email that's just for this trial, and we assure you that we will get back to you and connect with you. So let's start maybe with the video first about the experience of somebody who went through it.
Okay, 1 second, please. Do you all see the video?
My name is Amy Brooks. I'm from Tampa, Florida. In 2006, I was incidentally diagnosed with multiple angiomas while going through infertility treatments. In 2015, I began having seizures. In 2016, I had a craniotomy to remove the angioma that was in my left frontal lobe. Unfortunately, I continue to have seizures postoperatively. In June 2018, I ended up having a hemorrhage in my left frontal lobe. From the Angioma Alliance Facebook group, I heard so much about Dr. Awad in Chicago and became interested in hearing more about the Atorvastatin trial. So in September, I signed up. So I'm a survivor and I want a cure. I know we all do. And because I've had these two hemorrhages, there may be a chance that I'll never bleed again if atorvastatin provides good outcomes. This alone may improve my quality of life and even others. With this trial, there is only one way to find out. I feel so privileged to be a part of this trial. Dr. Awad and Christina, his nurse, have explained things in great detail to me, with my cognitive dysfunction, I have required repeated details to help me understand, but have completely understand at this point. I started the clinical trial 2 months ago. It is either the atorvastatin or the placebo, and only the pharmacist knows that. The process is very simple, though. You take the pill daily and document that you took it on a calendar, and you provide that to them. And then after a month of recording, you send the calendar and prescription bottle back via prepaid FedEx bag that they give you. Christina, she checks on you routinely to make sure things are going well and you have no problems or concerns. And then while on the trial for 2 years, you'll see Dr. Awad and his team members at least 3 times. Because I live in Tampa, which is greater than 200 miles, I receive a stipend of $500 to cover most of the cost. So I'm looking forward to continuing this process, and hopefully there will be others to be a part of this with. Thank you.
Thank you. Thank you so much for this very nice personal accounting of how things play out. So we will continue along this line. I want to let you know that the trial was launched late last summer, and the drug first became available, and therefore we started enrolling patients the first week in November. So we are now exactly 4 months into the trial. We have screened over 32 patients and have already enrolled 10 patients in the trial, plus 2 today. So we are 12 as of today, and this is ahead of the schedule. The trial is supposed to enroll 80 patients in order to complete, but we may have the answer with less patients depending on the effect of the drug one way or the other. So we will be looking at the effects of the drug as we go along, and we count the outcomes. So let us go next to Christina Paydad, who is the nurse who works with you every day and with me every day. Nothing is more important to Christina at work except CCM. So she gets the calls from you and the messages and connects back to you. And she's going to explain to you a little bit how to tell us whether you're interested in the trial and how the process plays out. Christina?
Yep. Hi, everyone. So the way it works, and you have this information in the brochures that were provided in the email, but there is a designated email address and phone number for this trial. So you can find that information there, and that's a good way to reach out to me. And I am very quick to respond. So what I'll first ask is some basic information, clinical information, and make sure that you have the most important criteria, which is that you've had a symptomatic hemorrhage within the last 12 months. And then there are a few other criteria I go over with for screening and eligibility. I ask you whether or not you are just looking for research, or if you are coming also to seek a clinical opinion from Dr. Awad. From there, we start gathering records together. Once those are received, I review them to see that everything is documented, if we have any other questions for you. And with your past medical history, we can tell what you might need when you come see us in Chicago. And then we set it up. Everything happens the same day. If you're coming for the enrollment, you're probably with us for maybe 2 to 3 hours. And then that's pretty much it. You get the MRI and you do the entire screening process. And then the drug is sent either home with you on that same day, or we mail it out to you. So I guess now Agnieszka will go over the consenting process for when you come in, and she describes more of the mechanics of the actual trial.
Thank you, Christina. Hi, everyone. First, I want to apologize for the background noise. I didn't quite make it home in time to call in. So Ms. Brooks did a fantastic job of explaining how this is going to work. So like Christina said, once you've had your records reviewed and Dr. Awad and Christina have determined you're eligible, you'll come into either the clinic or our other offices. I'm so sorry about the noise. And we'll go over the consent forms. So there's 2 of them. It's pretty lengthy process because there's a lot of information. There's a lot of visits to review. We go over the randomization process. So as Ms. Brooks said, again, we don't know what you're getting, whether it's placebo or atorvastatin. Only the pharmacy knows. So we'll go over that in detail to make sure everyone understands. And then once you've consented, if you agree to be in it, we start doing, like Christina said, the assessments. So there's a couple of neurological assessments we do. And then we also do your medical history to review anything we might be missing from your records. We draw some blood to make sure your liver function is okay, your kidney function, thyroid, etc. We put all that into a database that's run through Johns Hopkins, actually. And then the computer, if you're eligible, meet all the criteria, the computer spits out to the pharmacy which group you're in. And then, like she said, you'll either go home with the drug that day, hopefully, or if not, we'll send it to you within a few days. There's a call every 3 months with Christina just to check in with you to make sure you're not having any side effects from the drug, that you're feeling okay, no new symptoms from your lesions, etc. And then you come in to see us at 1 year and 2 years. We do all the assessments again, the MRI, etc. And then that'll be it for the enrollment, really. So I'll take questions after Dr. Awad, if that's okay.
This is great. Thank you, Agnieszka. That's exactly what we wanted to hear. So what happens during the next 2 years is obviously one of 2 things. Either you are getting better and there are no new symptoms, which is what we want, in which case we would do an MRI at 1 year and 2 years anyway to measure whether there has been more bleeding or less bleeding in the lesion and to see how the drug is having its effect. Heaven forbid, if you have any new symptoms that could be a rebleed or a seizure or any other symptoms, they get managed the way they get managed anyway medically. So if I'm taking care of you, you call me and we will talk to you about coming in and having a scan, etc. Or if you are continuing with your own doctor, you will do that. You will call your doctor and let them know. But we also obviously need to be aware and of your symptoms, so we can look and make sure it's not something the drug is having an effect and so forth. If a person has bled on the drug or the placebo, then we stop the drug and the placebo, and you make a decision with your doctor, whether it's me or whomever it is, as to what to do next, like you would otherwise. So it doesn't take away any of your other choices for the care of the CCM, including surgery or anything else. If at any time during the 2 years you change your mind and you decide, I don't want to do this, or I want to go ahead and have surgery after all, or whatever, even without a bleed, you have the right to do that, and you just let us know. Obviously, we'd like people who are committed to do the trial to enroll in it, so we do not waste the resources. But you are the most important person, and we want to honor your wishes at all times. The final thing I promised to tell you before we take questions is how does it play out between my opinions and your own doctors? Because many of you are not from Chicago and are not seeing me on a regular basis. So one of the first things that Christina will ask you is whether you and your doctors are interested in a formal second opinion, so that I am actually giving you an opinion on your CCM other than the research. If that is the case, we will work with your insurance to get authorization for that, and then you will be seeing me both clinically and for the trial. But if your insurance does not cover care outside your area, or you are perfectly fine with your doctor and don't need my opinion other than for the trial, that's fine. We work with outstanding surgeons and other CCM experts throughout the country and throughout the city of Chicago, with whom I will continue to stay in touch even as you are in the trial. So you can get into the trial and stay with your own doctor if that's what you want. A couple of differences will happen is that if you are coming to Chicago just for the research, then you will not be seeing me in the clinic. You will be seen in the research area, and you may only see the nurse. I'm available to you if there is a problem, but my job is not then to give you an opinion on your cavernous malformation. I'm just telling you how the trial is working with your lesion. If you are seeing me clinically, then we will schedule you in my clinic, and we will do both the research and the clinical visit. And we have no preference. It is your preference, and we have the ability to handle it both ways. Now, the payment for each of the 3 visits, the initial visit, the 1-year visit, and 2-year visits, is given to anyone, whether you're seeing me clinically or you're seeing me just for the research. So if you're in the trial, you will get given the $500 if you are from 200 miles or further away, or if you are from within Chicago, it is $50 just to help you with the trip to our place and the parking. And this is all you have to worry about. If you're seeing me clinically, we work with your insurance the way we usually do. If you're not seeing me clinically, there are no bills that you see related to this trial. So you will only get the stipend, the drug, and all the MRIs and everything would be covered under the trial. So that is what I wanted to tell everyone. I noticed there are a lot of questions by people about what brochure are we talking about. So I would ask you to look at the last email that was sent by Connie yesterday and today. You all received reminder emails, and at the bottom of the email there is a link to the brochure. You click on it and the brochure opens up, and you can download it or print it as a PDF. And if you cannot do that or are having a problem, just email us or call me or call Connie or Angioma Alliance. We'll get the brochure to you. All right, so I think I wanted to really do this mostly to answer your questions. So many people signed up because they had questions. So let's turn it to questions and answers, and I think this will give the information the best way. Who is going to be spearheading the questions? All right, Joanna, you are the boss.
There you go. I promise to speak slower. Question number 1. I always wonder if there is any possible chance that CCM can be genetic and passed on in my case. I only had 1 cavernoma. I know the genetic form typically only happens when multiple cavernomas are seen. Have you ever seen a patient with 1 cavernoma whose child had it as well?
Yes, so rarely the familial disease can have 1 lesion. However, this is so rare that the likelihood of that is lower than the chance of a false negative test, because even the genetic test can sometimes be false negative, maybe 1, 2, 3%. So if everyone was to go and get the genetic test with a single cavernoma and no family history, the chance of finding the familial disease is less than 1 in 1,000. So it's many, many times lower than the chance of the test being false. So it doesn't really make sense to spend several $1,000s doing that test when the chances are so low that it will be positive. However, if there is any clinical reason to suspect that your child or a loved one or your aunt or your mother had a CCM, then it does make sense for us to do the genetic testing. So the condition for doing the genetic test is either a family history or a question in the family or multiple lesions on the MRI.
Okay. Question number 2. Are there any studies and tests related to CCM I can participate in? Mine already has been surgically removed.
Yeah, so if your lesion is surgically removed, then you are one of 2 possibilities. Either it was completely gone and it's the only lesion, in which case you are cured. Congratulations. There's no study for you to do because you have no cavernous angioma anymore. However, if the surgery was incomplete, meaning there is still a cavernoma there, because sometimes surgery is done and not the whole lesion is taken out, or if you are familial and have other lesions that were not taken out, then we have to watch these other lesions. And there are many studies that are available, biomarker studies, microbiome studies. So check with the Angioma Alliance about what studies may be happening near your place. Currently, the majority of the studies are focusing on a lesion that bled in the last year, because that's really where we want to see a change. If we treat people who have not bled in the last year, the likelihood of seeing an effect of the drug is so small that we have to treat thousands of patients to see the effect. So don't feel discriminated against. That's what the only reason we're doing it in the people who bled. It's because we want to find the answer quicker. That's why we're doing it in the people who bled first. Thanks.
Okay, question number 3. I am curious to know the science behind how taste and smell hallucinations happen. So,
yeah. So there are parts in the brain where the cavernous angioma may be that are usually in the temporal lobe or in the frontal lobe, where if the lesion is causing seizures or is irritating the brain, it irritates the olfactory or taste circuits. So instead of causing a circuit that makes your arm shake or stop speaking, it makes you have a funny smell or a taste that is bad. So this is part of what we call partial complex seizures, where you have an altered awareness and the sense of something happening, but yet you do not lose consciousness. And these can be seizures from the cavernous angioma. So that's the biology.
Okay. Question number 4. What can I do in my community to raise awareness about cavernous angiomas? I'll answer that one. Get involved with the state ones that we, Tracy mentioned is already about, I think, 8 or so in the state, and we're trying to create more. But there's more to that question. I still can't get over the fact that I had seizures for 8 years without knowing they were seizures. Simple partials felt like panic attacks, and I never thought to tell anyone. How can we make cavernous angiomas a very common textbook medical issue that everyone knows about?
Well, that's what you guys are doing, right? Every one of you can make that. So we want you to tell your story to your doctor. Tell your doctor, what's wrong with you? I've been telling you this, listen to me. And they will listen, and this is how it goes. All of us, I think everybody on the call tonight is interested in doing what you want, and it's our job to, information can only be spread by information.
And with social media, it's very powerful. Question number 5. What are the effects of cannabis for CCM?
So on the lesion itself, the biology of the lesion may well be destabilized by the effects of cannabis. And we know this because of the effect of cannabis in other blood vessel diseases, like aneurysms and moya and so forth. We have not yet given cannabis to mice and see what happens to their cavernoma. Now, as far as the seizures are concerned, I know there is a lot of interest in using cannabis or its derivatives for seizures. Our approach to that is to have that overseen by an epilepsy doctor, because we don't want you to self-medicate and instead of stopping the seizures, get high so you forget about the seizures. We don't want that. We want the seizures cured and the disease treated. So our thought would be that, medical cannabis should be handled by a professional who knows about both cavernous angiomas, epilepsy, and cannabis. If you go to one that doesn't know the other side of this, you can go down the wrong road. And we have had many, many patients where excessive use of cannabis has been the only reason they bled. We cannot find any other reason for their hemorrhage. So I'm very, very cautious about telling people, yeah, do it and everything's gonna be fine, because we really don't know that.
Okay, thank you, doctor. Why isn't spinal CCM included in the trial?
Yeah, so spinal CCM is a little bit different because often when it bleeds, it causes pain, and sometimes it can cause pain and not bleed. So the symptoms of it are not as clearly correlated with the hemorrhage as the brain lesions. And also because sometimes for a lesion in the spine, even if it bled several times, we may watch it because it may be too dangerous to do something about it. So it's not the same decision tree as for the brain lesions. However, I promise you that if we see an effect in the other cavernous malformations, then automatically the drug will be used in the spinal lesion as well.
Great. When I exercise or am under stress, an optical migraine will trigger, lasting 5 to 15 minutes. Is it common for angiomas to become more active with increase in blood pressure due to exercise or stress?
It can happen. We have many, many patients that say that there are certain activities that cause either seizures or migraines. And obviously what you have to do is work out up to the level of those activities and not push yourself to the point where you are having triggered symptoms. The activity that's most likely to cause a bleed from cavernous angioma is the heavy lifting or straining. So if you are pushing and straining, like you are bench pressing 100 pounds or something like that, that is the type of activity that raises the pressure inside the cavernoma and can cause a bleed. So we usually tell people to exercise except for avoiding these extremes, and certainly if the angioma has bled before, to avoid these types of extremes until it has stabilized for a year or so, to make sure you're not triggering hemorrhage. We've had people where the only thing we found is that they've just started some very, very aggressive type of yoga where they're standing on their head in very vigorous ways. And we told them to just calm it down and go into a slightly calmer type of yoga, and they did fine. So you can push yourself to a point where you can make your lesion bleed. And don't do that. Life is worth it not to push to that point.
Okay. The next question is, my CCM was discovered incidentally 8 years ago, and he became a patient of yours. He had several MRIs since then with no evidence of a bleed. I know the study is for those who have had bleeds. I was put on Atorvastatin about 2 years ago to control my triglycerides. Has there been any indication as yet that it can prevent or reduce the risk of bleeds or in any way physically affect the CCM?
Yeah, so we were asked to actually look at all the patients in our registry. So we already looked at your case probably. And we looked at all the cases in our registry and counted the years they were on atorvastatin or other statins versus the number of years that other patients were not on that drug, and counted the bleeds per year of atorva or per year without atorva. And it looks like it does seem to be doing the right thing, meaning we have less bleeds in general in people who are on statin. But I would caution you, because the people who are on statin are not the same. They're a little bit older and they have high cholesterol, they have heart disease. So they may be in a different age group and different reasons for which they didn't bleed in the first place. So I would be very careful not to attribute that to the benefit of statin without a trial, but we can tell you it doesn't do any harm.
Okay. I have 3 CCMs in my brainstem and 9 throughout my brain. Could any of these, due to the location of CCMs, affect the digestive system, such as spasms in the esophagus or any digestive issues?
Usually not. The brain lesions do not usually cause these types of GI symptoms. So exactly not knowing where your lesions are, I cannot answer 100%, but I can tell you during more than 30 years of seeing CCM patients, the effect on the esophagus or the stomach from the lesion, a neurologic effect, is not common. However, if people have had a bleed, sometimes they cannot swallow very well, they have other issues. And also we know that people who have the familial CCM may have a different gut permeability issue, and they may be more vulnerable to inflammatory bowel diseases. So I wouldn't totally exclude the CCM as a reason, but it may not be the direct effect on the brain that you are thinking about. But we're trying to figure out these other mechanisms so we can maybe treat the gut of CCM patients better. We have several people in the laboratory working on that.
Very good. Would the results of the study be applicable to sporadic calves as well?
Yes. So the mechanism by which a CCM forms at the end is the same. The same molecules are screwed up, whether it's a sporadic lesion or a familial lesion. The only difference is that the familial lesion, you have the seed throughout the body, so you can have more lesions. But the sporadic lesion in that one spot is identical, including all the molecular triggers to the familial lesion. So what makes it bleed and what makes it stop bleeding should be the same. And this is why both sporadic and familial cases who bled within the prior year are being studied in the trial.
Okay. Does an anti-seizure medicine or Theraprox preclude participation in the trial?
No, both are okay.
Okay. I have a cavernoma at T8 and it causes radicular pain and neuropathy in my left leg below the knee and in the foot. I recently had a heart attack and as a partial result had a drastic reduction in blood pressure from 127/78 normal to 110/60. My pain and neuropathy have reduced more than 90%. Do you know of any studies or papers that correlate blood pressure and symptoms of cavernomas?
We know that when blood pressure is high, the cavernomas can bleed. But I think other than that, effect of blood pressure on pain is harder to explain. But it could be that your lesion was going to settle down anyway, and that this may have happened incidentally, meaning, people who have something, they did something the day before, and I always want to say it happened because of what I did the day before, but sometimes the things were going to happen anyway.
Okay. If this drug trial is successful, could this drug be beneficial to a patient that has had a lesion removed, but there is a possibility that it could grow back? Yeah,
these are what we call secondary effects. So depending on how powerful the effect of a drug, its use in the clinical community will then become more acceptable. So people will widen the indications. So initially in a trial, the indications are narrow, so we find the answer. But then ultimately, if the drug works and doesn't do any harm, people will use it for other applications.
Okay. This is a little long. I have a CM in the thoracic spinal cord. Its size is more than 2 centimeters and multiple ones in the brain. I found it 10 years and have felt symptoms last year. The doc from Heidelberg told me I can have a trial for propanol, I guess. I have taken it for 3 months, but the symptoms didn't change. Should I continue to take the medicine in the future? I think this is...
Yeah, so propranolol, there is no trial in America for propranolol. They're just trying it. There is a difference between trying it and the trial. Trying it, meaning the doctor is just giving you the medicine to see what happens. And as it turns out, we have as many people who say it didn't work as we have people that say it works, and we don't have an answer because there is no trial. A trial is done with half the people on it, half the people without, and not knowing. Not the doctor knows, not the patient knows, so we can be sure that it's the drug doing the effect, not our wishful thinking. So propranolol is being done in a trial in Italy. That's the only place where it is being done in a trial. It may be used in a trial in the US if we find an effect in animals or a more promising effect in human beings. But until then, we don't have the same evidence for propranolol trial that we had for Atorva. So it's not like we liked one but not the other. We had to go after the one that had the scientific premise. And propranolol is just a try and see. In fact, it's enough of a try and see that you can do whatever you want, you and your doctor with it. It probably is not going to make a difference. And you are still eligible to participate in the Atorvastatin trial if you bleed.
Okay. Is statin useful for the cavernoma in the spinal cord?
We do not know. If it works in the brain, if we find that it works in the brain, then the answer is yes. But until now, we don't even know that it works in the brain. So let's find the answer first, then we'll use it for the spinal cord too.
Okay. Who will be the most suitable neurosurgeon for the intramedullary cavernoma in the America and Europe?
The best neurosurgeons. So you have to look it up and follow your neurosurgeons. I'm not going to advertise for individual neurosurgeons tonight.
Do you think the cavernoma surgery for brainstem is similar to the intramedullary surgery?
It all depends, really. It depends on the case, on the size of the lesion, how close it comes to the surface, etc. So those are the questions you ask to your surgeon.
Okay, very good. What are your outcome measures? Fewer bleeds, shrinkage. What are you measuring that the statin is doing?
That's an excellent question. So the primary outcome measure, this is the one where the trial succeeds or fails, is the amount of leakage in the lesion. So which we are measuring on MRI in a very special MRI. That's why you come to Chicago. I want to meet you and see you, but the reason you come to Chicago is really to get that special MRI. Currently, we are trying to validate the same MRIs at other centers of excellence like Mayo Clinic, UCSF, etc., and New Mexico. But we are not quite there yet. We are in a study that will take 2 years. It's called trial readiness, where the same MRI that's done in Chicago will be doable at other places. But until we get there, I'm afraid the only place where we can measure it accurately, and we know we're measuring accurately, is in Chicago. So that's why you have to come to Chicago to have it done. So we are actually measuring how much bleeding there is inside the lesion, no matter how you're feeling. Even if you're feeling good and there is bleeding in the lesion, we don't like that. So we're going to measure that. Conversely, if the statin is having an effect and decreasing bleeding in the lesion, even if you're feeling fine, it's going to be a better thing that the lesion has been stabilized. So the primary outcome is the amount of leakage in the lesion. The secondary outcomes, we're counting bleeds. So we want to know, are there more bleeds in people on statin or less? And if there are more bleeds in people on statin, we are going to stop the trial.
Okay. And the next question says, at the end of trial, do they reveal to the patients if they were taking the drug or the placebo?
What's the rule on that, Agnieszka?
Yes, if after the results are in and all the follow-up is done, we can reveal what each person was taking.
Yeah, so until then, the only time we break the blind is if there is a life-saving reason to do that. So if there is some unexpected complication that we find, I have the right to make the decision to break the blind. But I won't do it just because you're begging me. You're not allowed to do that.
What opportunities are there for CCM2 patients? My husband and my 50-year-old son have too many lesions to count on their MRI.
Is it CCM2? Is that right?
CCM2, yeah.
Yes, it's unusual why you have so many lesions with CCM2. Most CCM2 patients have less lesions than the others. But of course, your case illustrates that you could have any genotype and have a lot of lesions, or have the worst genotype, CCM3, and not have many lesions. So there is a lot of variation. We do not know why you in particular have more lesions. You may have been vitamin D deficient. You may have had the wrong microbiome at different times in your life. We are figuring those things out. But the nice thing is that we are doing this study called the biomarkers study, that if you are getting your care in Chicago, we also send blood, we ask your consent and we send blood work. And what we are trying to do is find out what are the markers in the blood that can predict which patients will form more lesions or less lesions, which ones will bleed and will not bleed. And we're trying to figure it out. So stay tuned with us.
Okay. I am in Australia. Is there any way to participate in this trial informally? Assuming this drug is readily available here, would it be okay to take it as a precaution?
I would not recommend that, because you are not going to be having the measure of the outcome. So you would be taking it and not knowing whether it did harm or good. And if you bled when you're on it, we're not counting you the same way as the others. So your case is as likely to be harmed by the drug as helped by the drug. So I recommend you stay tuned till the results are there. Foreign patients who are coming from out of the country under some conditions can enroll, but they have to be able to have an address in the United States that we can ship the drug to, because we cannot ship drugs across international borders except to Canada. And therefore, we have had a patient from Brazil, for example, but she works in the United States and she comes and has an address in the US. We also want to be able to see the patient at 1 year and 2 years. So, and the last thing is $500 is not enough to come all the way from Australia. So it's going to be a hardship on you. But if you are able to participate in the trial and want to, yeah, we'll work with your doctor. They speak good English in Australia and they're great doctors. So we'll do just fine.
And you just answered the next question was, is this study for the US only? So the next question, my cavernoma has not had a bleed that I've known of. However, it is growing. Over the past 6 years, it has grown, almost doubled in size. Would the statin possibly help me in the future to stop mine from growing?
So if the growth is symptomatic, then it is the same as a bleed. So we can count it as a CASH. So CASH is a hemorrhagic growth or bleeding. So if it has grown, that means there is new blood in it. That's how cavernomas grow. So the main question is going to be, is it symptomatic or not? If it's symptomatic, then you are eligible for the trial. If it's not symptomatic but growing, then you should talk to a surgeon about taking it out.
How does the statin work on the CCM? Are you thinking it impacts the muscles on the blood vessel walls?
No, because the blood vessel walls of cavernomas have no muscle on them. So the blood vessel walls of cavernoma are very thin, and they're only made of endothelial layer, and that's it. That's why cavernomas bleed. So the way statin works is on the junction between the endothelial cells. So the endothelial cells are zipped together to keep the blood vessel from leaking. And in the cavernoma, that blood vessel leaks. The statin has been shown to make the blood vessel zipped again, so it is less likely to leak. And it works via a mechanism called Rho kinase inhibition. So when the blood vessel zips out and it leaks, Rho kinase is increased. And when we inhibit Rho kinase, the endothelial cells are more impermeable. They're less likely to leak.
Okay. Do you advise pregnant women to deliver via C-section or vaginally?
Okay, I'm going to turn this to Christina, who answers this question all the time. What do we do, Christina? Can we hear her?
Yeah. So if your cavernoma has been proven to be stable on MRI, then there's not really any indication for you to have a C-section. We recommend that you that we all coordinate with a high-risk OB to determine what other OB precautions you would need to take. But as far as a cavernoma, if it has been stable with no recent symptomatic events, then normally it's totally fine to have a normal vaginal delivery.
So that is the evidence-based answer, and that's what we practice, and that's what the guidelines say. So if you are worried, then in your second trimester of pregnancy, we can do an MRI without contrast, which is totally safe, without dye, and tell you that your lesion is stable. And if it's stable, we clear you for vaginal delivery. If it has any changes during pregnancy, then we'll send you towards C-section.
Okay. Even though I wear a hat going outside in the severe cold, the temperature shift sends a brain freeze that is so intense. Can anything be done about this?
No, I guess move to Florida.
We'll welcome you here in Miami.
I'm really sorry, but I don't think so, because I don't know exactly what's the relationship of those symptoms to the cavernoma. So I would ask you to bring it up with your doctor.
This is a long one. My father is part of the trial. He was also previously on the beta blockers, the propranolol, the one I can't pronounce. He recently stopped taking this medication as he was on it to prevent high blood pressure in high stress situations. He was experiencing extreme fatigue, but yet only able to sleep 4 hours a night. Have you heard of this side effect?
Yeah, some people on propranolol said that they are tired with it and they feel better without it. If he is currently on the trial of the statin and is feeling tired, he should let us know. And we will talk and we will do the blood test to make sure it's not a statin side effect.
All right. She continues with, I don't know if you can answer this, but within a couple of days of being off of it, he seems to have energy back and sleep is still to be determined. Would you advise he remain off the medication? Is there any reason he should go back on the medication?
We're talking about propranolol, is that what you? Yeah, I mean, whoever started it should tell you whether you come off of it or not. You know, I would not have started it in the first place. So if you have side effects, yeah, come off of it and you'll probably be the same with it or without it.
Okay. Next question. Why are there times that I get symptoms? They put excessive dizziness, migraines, slurred speech, but my MRI is fine. I have too many to count and one removed from the brainstem in 2014. So I guess the question is they're not seeing anything in the MRI, but they're having symptoms.
Well, they are seeing on the MRI that you have a lot of lesions and that you also had prior brainstem surgery. So as you know, and as everybody who has been through this knows, that the lesions have an effect on you. They can make you more emotional, they can make you more tired, more intolerant of cold and heat. And previous surgery and previous brainstem events and strokes and seizures can all alter our mood and our sensations. So I think these are due to the CCM, but the reason the MRI doesn't show a change is because, thank God, you are not bleeding actively or forming new lesions. But that doesn't mean the lesions are gone.
Do you do video appointments for a second, third, or fourth opinion?
I don't do that because I think those of you who have come and seen me realize that the experience of talking and understanding a little bit your life and what brings you up to this point makes a very big difference in the decision we ultimately make. I know you could say, well, maybe you can do the same on the video or whatever, but I'm not going to be able to do the proper MRI. I'm not going to be able to draw your blood. I'm not going to be able to. So probably those opinions will be shallow and will not be as meaningful as patient care opinions that we give. And that's why I rather use my time for the more useful type of consults. I realize many people want to show me their MRI and ask me what I think, but that's kind of, you know, shallow, and I can be wrong because I don't know you very well. And I don't want to give wrong opinions. So this is why I'd rather use my time, which is limited, to take care of the people who want to be taken care of. Now, occasionally, people will ask, is my lesion surgical? So that I come and talk with you about the option of surgery, or is my lesion should not be touched by surgery? And we can look at some of this and give you a quick answer, but it's not going to be a final and official opinion.
Okay.
Joanna, can I interrupt real quick? Can you check on the chat? There are some trial-related questions, if we can get those, just because we're
getting close to the end of the webinar. Yeah.
So if we can get there's a few trial-related ones, if we can hit those before the.
Okay, if you want to read those, because I was just going through the question and answers. So if you read any that you think should be answered, please do that. Or I guess I'll ask the next one. How do you determine if a CCM is considered stable?
Well, it's stable if it has no growth, no bleeding, and no symptoms. So we make that decision every time we see a patient, and I tell you if your lesion has been stable or not stable.
Tracy, you want to ask her?
Yeah, hold on, I'm scrolling. Go ahead and do another one till I got you one ready.
Here, how conclusive are the studies that link CCMs and vitamin D?
Yeah, so the study is not a trial, was never a trial. So the study took a large number of patients, and we looked at their vitamin D level. And we looked at how aggressive their cavernous malformation had been in their life, okay? And we asked whether vitamin D can predict if you are going to bleed next year, or whether it could predict whether you could bleed last year. And that didn't correlate. But what was very, very strong is that vitamin D deficient patients whose level was less than 25 were many times more likely to have more lesions if they are in the same family, or more bleeds over time in their life. So the way we explain it is because of the pro-inflammatory effect of vitamin D deficiency. So vitamin D, it kind of chills out the inflammatory activity in the body, and that's how it does some good for CCM. However, it is not an index if you're gonna bleed next year or if you bled yesterday. It more tells us about your activity over a long period of time. So there is no downside to taking it. I tell people, if you hate it, don't take it. But it's not a drug we're ever going to spend $5 million to do a trial, because everybody will cheat and go and take it, which is fine. You know, so take vitamin D, and there is no downside to doing it, but there could be a downside if you don't.
Great. Okay, next one. How long will patients be followed on the trial? And if the trial is successful, what is the process after the trial is completed? And when would it be available to children?
Yeah, so all of those are good questions. Maybe this will be the last questions on the webinar because we're getting to that time. But I think the trial is designed to follow each patient for 2 years. And so each of the 80 patients enrolled will be followed for 2 years, and each year will be counted, whether there was change in it or a bleed in it. So we'll have a number of lesion years on statin and the number of lesion years off statin, and the comparison will tell us what the outcome is. However, the trial could be stopped early if we have too many bleeds on statin, then we'll stop it. Or if we have too many bleeds off statin, we'll also stop it because it means statin is working. Okay, and we want to declare that information. Also, we will stop the trial if it looks like it's futile, meaning after we enroll 40 patients for 2 years, it looks like there is no difference between the two whatsoever, zilch, zero. Then there's no point wasting your time for another 2 years, okay? So we'll do these analyses, we call them interim analyses, and if there is an answer, we're gonna broadcast it to the world and decide what the next step would be. Then whether you use it in children or not will then be based on at least some evidence that now we don't have. So you and your doctor will be able to make the best decision.
Okay.
So I realize that there are a lot of questions that went unanswered. So I'm going to make a suggestion that if we can categorize these questions into like 4 or 5 that everybody is asking, send them to us. And maybe Joanna and everybody at Angioma Alliance can say, well, these are like the 5 to 10 big questions that everybody really wanted to know and we didn't get to it. And we're happy, Christina and Agnieszka and myself to, you know, kind of send you an answer that you can share with everybody. Great. And the other thing we promise is that we should be doing this as we go along. So maybe every 3 to 6 months, we should do this. And we'll give you both an opportunity to ask questions, and we'll give you update on what we're learning from the people who are enrolled and how things are going.
Great, thank you.
Thank you really, everybody. I really want to thank also Agnieszka and Christina because they were working all day in the CCM clinic. We enrolled 2 patients in the trial today. It was a lot of work, and we went home to do this, but that's because it is really worthwhile. So, and thank you all for doing this with us.
And do you want to share Christina's contact information if they are interested in enrolling in the trial?
Yes, so it's ATCASH. ATCASH at uchicago.edu. Okay, so it's a very simple email.
Sorry, it's actually, it's UC Hospitals.
Oh, uchihospitals.edu. Okay, let's see that again. It is on the brochure if you open the brochure, but it is at cash, ATCASH at uchihospitals.edu.
Perfect.
And you can go to our website, okay, and it's there also. So all the contacts are always there. It's hard not to get a hold of us. So just call and do whatever, and we'll get to you.
Right. Thank you very much, Doctor, and everyone for coming and participating. As Dr. Awad said, we plan to do this every 3 months. So most likely come May, we'll hope to have another webinar with an update, and he'll be answering questions as well. I'm sorry if your question wasn't answered. I know there were a lot, but we'll be sending him, you know, the 5 most common. So expect an email from us in the next few days with those answers.
And maybe next time we'll talk less and we'll answer more questions.
There we go.
Thank you, everybody.
Thank you, everyone. Have a good night.
Bye-bye. Thank you, Joanna.
Thank you.